So I would first like to thank the Karnataka Pulmonologists Association for giving me this platform for discussion. A warm good evening to all the viewers who are catching us live this evening. The topic for today's discussion is on pulmonary stretch receptors. So I would first like to begin with the ancient German tale, which became very popular after a play in the early 1900s named as Ondine. So this Ondine was a beautiful water nymph who fell in love with a mortal man. Later, she realized that he turned to be unfaithful, and just like any other woman would react, she was totally outrageous. So she just cursed him. There are a lot of versions related to these curses, but all of that is related to his breathing. So he was cursed that his automatic or the involuntary control of breathing is lost. So he had to stay awake all the time to make sure he's breathing. So at one point of time, he really got exhausted, fell asleep, and died. So this was very popularly known as the Ondine's curse. And in today's scenario, this same thing is a sleep related breathing disorder known as central hypoventilation syndrome. So we have very few cases worldwide, around 1,000 of them. So they can be either congenital or due to any brainstem injury. So here the involuntary control of respiration is lost. Hence the patient or the child has to always be on a ventilator, or tracheostomy is a must. Of late, we have a new technique known as a biphasic curious ventilation, which can replace the tracheostomy. So now we know that how important our involuntary control of breathing is. So that is what I'll be discussing in the regulation of respiration briefly, so that we understand how these pulmonary stretch receptors would be working. So this is the overview of today's presentation. So breathing is a beautiful process which happens involuntarily even when we are asleep. But however, we have a voluntary control also. So the involuntary or the automatic control of breathing is controlled by the centers in the brainstem, that is the pons and the medulla, whereas the voluntary control is by the cerebral cortex. We also have various chemical factors like partial pressure of oxygen, carbon dioxide. Through various chemoreceptors, they also regulate respiration to make sure that they maintain their levels constant in the blood. So apart from these two mechanisms, we also have a non-chemical regulation, that is the reflexes which originate from the airways. These reflexes basically modify the neural mechanisms. So regulation of respiration mainly consists of genesis of a spontaneous respiratory rhythm and controlling the rate and depth of respiration. So this is by sensory inputs received from the mechanoceptors, chemoreceptors, the various receptors in the chest wall and the lungs, which relay in a central station, that is our medullary center, and the effect is brought about through the spinal nerves acting through the muscles of respiration. So as I told you, the neural regulation has an involuntary control by the centers which are present in the brainstem, that is the pons and the medulla, and the voluntary control is by the cerebral cortex. So the medullary and the pontine centers are again further divided. So medullary centers are basically responsible for generating a spontaneous respiratory rhythm, whereas the pontine centers, they control the rate and depth of respiration. So we have two group of neurons in the medullary center, that is the dorsal respiratory group and the ventral respiratory group. And in the pons, we have two important centers, which are the apneustic center and the pneumotaxic center. So medullary respiratory center, as I told you, the main function is to generate a basic respiratory rhythm. So we have the dorsal respiratory group of neurons, which are located bilaterally around the NTS. They are mainly the inspiratory neurons. So these neurons spontaneously fire discharge initially at a lower frequency, and gradually their frequency increases. So this forms a ramp-like signal, which is known as an inspiratory ramp. And after 2 seconds, there is sudden stoppage in this firing, which ceases the inspiratory ramp. So this is when expiration, which is a passive process, occurs. So DRG mainly consists of inspiratory neurons responsible for an inspiratory ramp. Whereas the ventral respiratory group of neurons, they have both inspiratory as well as the expiratory neurons, but however, they are inactive during quiet breathing. So when do they play a role? That is mainly during forceful respiration, as in case of exercise. So pontine centers, they basically regulate the rate and depth of respiration. So we have the apneustic center, which is present bilaterally in the lower pons. This basically prevents the switch off of the inspiratory ramp, meaning the inspiratory ramp continues, thereby causing an increase in the duration of respiration. So this is known as apneosis. The temporary arrest of respiration in inspiration is known as apneosis, but this normally doesn't occur because the apneustic center is inhibited by another center known as a pneumotaxic center, which is located in the upper pons. So as I told you, we have the dorsal respiratory group of neurons, which are mainly inspiratory neurons, which spontaneously fire, lead to an inspiratory ramp, and the ventral group of neurons, which contain both inspiratory and expiratory neurons. Both are present in the medulla. They are responsible for spontaneous generation of a respiratory rhythm. So in the pons, we have apneustic center, which leads to apneosis, but this is normally inhibited by the pneumotaxic center. So this is the one which controls is responsible for involuntary or automatic control of breathing. So next we have the chemical regulation. It's mainly by changes in partial pressure of oxygen, carbon dioxide, and H plus ions, which are basically detected by these chemoreceptors. We have peripheral chemoreceptors, which include the carotid body and the aortic body. The carotid body is present at the bifurcation of the common carotid artery, whereas the aortic body is present in the arch of the aorta. We have central chemoreceptors in the ventral surface of the medulla. So the peripheral chemoreceptors are more sensitive to fall in partial pressure of oxygen and fall in blood pH, or increased H plus ions are detected by the peripheral chemoreceptors, whereas the central chemoreceptors are more sensitive to changes in partial pressure of carbon dioxide. Mostly an increase in carbon dioxide partial pressure of around 50 mm of Hg stimulates these chemoreceptors. So they send impulses to the brainstem, which acts on the muscles of respiration, increases the rate of respiration. So thereby, if there is hypoxia, there is more of oxygen which is taken in, so which gets back the partial pressure of oxygen to normal levels. If there's increased carbon dioxide, it causes a carbon dioxide washout. So somehow all these levels are tried to be maintained at constant levels in the blood through this chemical regulation. So the third important mechanism is our respiratory reflexes, which basically modify the neural mechanisms. So they are important for fine tune of breathing. So basically they play a role while protecting the lungs from various environmental insults. So we have three types of receptors which basically modulate these respiratory reflexes, which include the slowly adapting receptors, also known as pulmonary stretch receptors, which is a topic for today's discussion. There are also rapidly adapting receptors. Sometimes they're also known as rapidly adapting pulmonary stretch receptor. The third type is the C fiber endings. Now we will discuss all the three slowly adapting receptors more in detail. So what we have to remember here is these receptors are nothing but afferent nerve fiber endings. So the slowly adapting and the rapidly adapting receptors are myelinated afferent nerve fiber endings, whereas the C fibers are unmyelinated. And the second important difference is the slowly adapting receptors are present in the smooth muscle of the airway, whereas we have this rapidly adapting and the C fibers are present in the airway mucosa. So we'll again discuss more in detail about the same. So slowly adapting receptors are also known as a pulmonary stretch receptors. They adapt slowly to stretch imposed on them. So the best way to understand this is, for the first time when we sit, we are able to feel that we are sitting, okay, because the touch receptors are stimulated. But after a few minutes, we forget or we don't get that input that we are sitting. Or another example is when you're wearing the clothes. The moment you wear the clothes, you can feel that there is some stimulus which is touching the body. But after a point of time, your adapters, the touch receptors get adapted. So you don't feel so conscious that there is something which is stimulating these receptors or something which you're wearing. Meaning these receptors get adapted, the touch receptors. But imagine I just prick with a pin. Initially you feel the pain. I continue to prick more and more. you still continue to feel the pain because these receptors don't get adapted. Okay, so they don't get adapted. So just like how these receptors are there in the skin, we also have the same kind of receptors in the lungs also. Okay, so these slowly adapting receptors initially respond to stretch stimulus and they continue to respond even when the lungs are stretched. Whereas the rapidly adapting receptors, they initially respond. After a point of time, if the stretch continues, the rapidly adapting receptors do not respond. Okay, so the slowly adapting receptors are something like a pain receptors which are continuously firing whenever there's a stimulus. So the location, they are present in the smooth muscle of the airways, and the stimulus for the stretch receptor is the depolarization of the stretch causes the depolarization of these receptors. So this is the structure of a slowly adapting receptor. So as we can see it is present in this, the slowly adapting receptor is present in the smooth muscle, whereas the rapidly adapting receptor or the C fibers, they are present in the airway mucosa. Okay, so coming to the structure of this slowly adapting receptor or pulmonary stretch receptors, they resemble the muscle spindle which is present in the skeletal muscle. This basically controls the length of the muscle fiber. It prevents the overstretching of the skeletal muscle fiber. So this structure is similar to a muscle spindle. It is around 100 micrometer in diameter. As I mentioned, these are myelinated afferent fibers which are present in the smooth muscle of the airways, mainly the larger airways like the conducting airways like the trachea, bronchi, bronchioles. The terminal branches of these, they wrap around the smooth muscle and they are sometimes overlapped by collagen fibers. So I repeat about the structure. They are around 100 micrometer. They are myelinated afferent fibers. They are located in the smooth muscle of the airways. The terminal fibers wraps around the smooth muscle, sometimes overlapped by collagen fibers. They resemble the structure of a muscle spindle which is present in the skeletal muscle. So coming to the types of pulmonary stretch receptors, we have two classifications. One is based on their location. Okay, so based on the location, we have two types, the intrathoracic or the extrapulmonary receptors, and second is the extrathoracic or intrapulmonary receptors. So in a simple way to remember, it is extrapulmonary, meaning outside the lungs. These are the predominant types of pulmonary stretch receptors which are present extrapulmonarily, meaning in the trachea, bronchi, and the bronchioles. So they have a rhythmic pattern of discharge, meaning they have a mounting discharge during inspiration and a declining pattern during expiration. The extra thoracic or the intrapulmonary that is present in the alveoli, they are very few in number. They have an irregular pattern of discharge. So most of the time when we say pulmonary stretch receptors, it refers to the extrapulmonary receptors which are present in the trachea, bronchi, and the bronchioles. Second classification was proposed by an Indian scientist, Pental, which is based on the discharge activity. They have classified it into low threshold receptors and high threshold receptors. So low threshold slowly adapting receptors, they discharge during expiration, whereas high threshold, they are silent during expiration. So this is one of the recording in an anesthetized rat. So whenever we take a recording of the respiratory movements, what we have to remember is the upward slope represents expiration and the downward slope is inspiration. So here we have the discharge of the receptors. Below that we have the movements which are recorded based on inflation. Okay, so as we can see here, just before inflation, okay, just before inflation, that is, there is expiration. During expiration, the low threshold receptors are firing, whereas here during expiration before inflation, there is no activity of the receptors. So low threshold receptors mainly fire during expiration, present in larger bronchi, and predominant pulmonary stretch receptors are of low threshold. So whenever we say pulmonary stretch receptors, mostly they are extrapulmonary receptors present in trachea, bronchi, bronchioles, and they are low threshold receptors. Okay, the very few of them are intrapulmonary and of high threshold type. So whenever the lungs expand to a certain volume, there is a reflex mechanism to protect overstretching. So there is a rest of inspiration and expiration begins, meaning there is an inspiratory off switch which is activated. So this controls tidal volume and the time required for inspiration. Similarly, the expiratory discharge controls the expiratory time. So when the expiratory time is prolonged, okay, the number of cycles of respiration is reduced, okay, meaning the breathing frequency is reduced when we increase the expiratory time. So all this is controlled by our pulmonary stretch receptors. So coming so far, we saw the types of receptors, pulmonary stretch receptors, where they are present, the structure of them, and also the classification of the pulmonary stretch receptors. Now coming to the properties of pulmonary stretch receptors. They are slowly adapting end organs. So I told you what we mean by slowly adapting, meaning irrespective of whatever the stimulus is, it is responding. I mean the stretch stimulus in this case, meaning this, if you can see in the diagram, this is the normal lung. There is a little firing of these receptors when the lungs are inflated. When there is suddenly stretch, there is a rapid firing of the slowly adapting receptors. And when the lungs continue to be in a stretch state, still these receptors continue to fire, meaning they don't come back to the basal firing rate. They continue to fire a little more than how much they were firing at the basal level. So what we are trying to say is, even these pulmonary stretch receptors respond not only to the rate of stretch but also to sustained stretch. Stretch itself is a stimulus, and the rate of change of stretch is also a stimulus. Okay, so even when there is sustained stretch, there is a activity of this receptors which is seen. That is what we mean by slowly adapting. Second important property is larger diameter. As I mentioned, they are around 100 micrometer. They are dynamic. Dynamic meaning there is discharge activity, which is mounting pattern during inspiration, declining pattern during expiration. There is serial discharge of these impulses when stretched, meaning they respond to rate of change of stretch as well as to stretch itself. And as I told you, they are low threshold receptors. So next important property is, these receptors are at okay, some pulmonary stretch receptors are tonically active at FRC. FRC is functional residual capacity, meaning the volume of air left in the lungs, present in the lungs after normal tidal expiration. Okay, so at this level we have only very few receptors which are actually active. Then when are all these receptors active will be the next question. So these receptors in humans are active only when the tidal volume reaches at least 3 times the normal tidal volume, meaning around normal tidal volume is around 500 ml. So only when the tidal volume reaches up to 1 or 1.5 liters, then only these pulmonary stretch receptors fire. So when this might happen, it's during conditions like exercise. So these receptors are active for all practical purposes only when the tidal volume reaches more than 3 times the normal eupnic tidal volume, that is around 1.5 liters. But however, they're sensitive, they are firing at FRC at residual volume up till vital capacity. The receptors are active. It's just like your mobile signal. When you have a single line, no point. You can't even make a call, you can't even access net. There's no point in having that little network. Only when there is full network, that is when you can use your phone either to make a call or access net. So what I'm trying to say is, when some SARs are fired, no point. There's no action potential generated which is capable of bringing any response. So maximum response or response itself is brought about only when maximum SARs are active, which happens only when tidal volume reaches 3 times the normal eupnic tidal volume. So as I've been telling, the stimulus is stretch. So these are mechanosensitive receptors which respond to stretch, rate of change of stretch as well as stretch itself. And they're insensitive to chemical irritants for all practical purposes. We have few journal articles which say that they may be responsive to change in chemicals or also certain chemical stimuli, but for all practical purposes, we will consider these are mechanosensitive receptors which are insensitive to chemical irritants. Okay, so hope so far it's been clear. So we will go in detail about one property of this receptor which plays a role in Herring-Brewer reflex. So this is also known as a pulmonary stretch reflex. So Carl Herring, a German physiologist, and his student Joseph Brewer. So both of them did an experiment on a rabbit. They artificially inflated the lungs of the rabbit, and they saw that when they inflated, suddenly at one point of time, there was an automatic stimulation of expiration which occurred. Okay, and that inspiration ceased at that point. And when they started deflating the lungs, they suddenly saw that there was automatic beginning of inspiration. So they named this as a Herring-Brewer reflex, and this was a protective negative feedback mechanism which prevents either overstretching or the collapse of the lungs. Okay, so this was very significant because this was the first negative feedback mechanism which was actually explained in physiology as early as in 1868. So this Herring-Brewer reflex is a inhibitory protective reflex. It's a protective reflex. It is inactive during normal breathing, so they don't play any role in regulation of tidal respiration. So they come into a picture only when there is overinflation or deflation. Okay, so normally they don't play much role in the normal regulation of tidal inspiration, so they are inactive during quiet breathing. This is an inhibitory protective reflex. So this is strongest in rabbit and weakest in humans, and the afferent impulses from these receptors are carried to the vagus nerve. Okay, so if we cut off the vagus, definitely this reflex is abolished. So that was actually proved in one of these experimental evidences. So we can see the first thing is the EMG recording in the diaphragm and the tidal volume. So the first graph is in a normal subject, second in a lung transplanted individual. So what we have to observe here is, in this is the tidal volume. Due after inflation, the upward curve, I've always told you, represents expiration in most of the stenography or the recordings of breathing movements. So whenever there is passive inflation of the lungs in normal individuals, once it reaches around 1.5 times the tidal volume, suddenly there is stoppage of inspiration and beginning of expiration. So that is what we see in normal subject. That is what is our Herring-Brewer reflex. But in lung transplanted individuals, normally only the breathing pattern is not regular because the vega is bilateral vagotomy. This is in a bilateral lung transplanted individual where both the side vega are cut. So normally also there is no regular breathing pattern, and on inflation also we do not see the typical effect which we have seen in a normal subject. So this shows that vagi carry impulses from these receptors. So if they cut, the Herring-Brewer reflex is abolished. So there are two types of Herring-Brewer reflex: Herring-Brewer inflation reflex and Herring-Brewer deflation reflex. So Herring-Brewer inflation reflex, as I've told you, it is a protective reflex. It's a negative feedback mechanism. So they play a role only when there's overinflation, meaning tidal volume reaches 3 times the normal eupnic tidal volume. So in neonates, they are normally also active and they are mainly responsible for the normal rhythmic breathing pattern, unlike in adults. Even in infants, they control tidal volume and they are responsible for normal respiration. Then why not in adults? Why not they play a role during normal respiration in adults? That is because of the postnatal habituation of this Herring-Brewer reflex. So in adults only it is functional when tidal volume is more than 3 times the eupnic tidal volume, whereas in neonates and infants, this Herring-Brewer reflex is seen during normal tidal inspiration and also is responsible for normal breathing, rhythmic breathing pattern. So what exactly is Herring-Brewer inflation reflex? So whenever there is lung overinflation, like the eupnic volume is around 500 ml. So when tidal volume reaches around 3 times, meaning 1.5 liter. Most common physiological condition when this happens is during our exercising. So when there is overinflation, there is stimulation of these pulmonary stretch receptors, also known as slowly adapting receptors. So once these receptors are stimulated, they fire action potentials. Okay, this is carried through afferent nerves, which is the vagus nerve, relays in a center, that is our medullary inspiratory centers. Now this medullary inspiratory centers, because there is overinflation, they send inhibitory impulses through the efferent nerves, that is the spinal respiratory motor neurons, that is a phrenic nerve supplying the diaphragm, and the spinal nerve supplying the external intercostal muscles. Meaning here the inspiratory off switch gets on, or I can simplify it to say that the inspiratory ramp is stopped. Okay, so once inspiratory ramp is stopped, what happens? There is inhibitory signals. Whatever stimulus was going to the respiratory muscles of inspiration, those signals are inhibited. So there is inhibitory signals to the diaphragm and external intercostal muscles. So once these muscles relax, there is abrupt termination of inspiration. So we all know expiration is a passive process, meaning once inspiration is stopped, automatically there is expiration which begins. So whenever there is overinflation of the lungs, the if net effect brought about is abrupt termination of inspiration followed by prolonged expiration. So this prevents further inflation of the lungs and further damage to the lungs. So that's why we call it as a protective mechanism, and it is a negative feedback mechanism. When there is overinflation, it tries to reduce the volume. So when there is less volume, it tries to increase the volume. So this is a negative feedback mechanism. So if I have to trace a reflex arc of this Herring-Brewer inflation reflex, what was the stimulus? It was overinflation of the lungs, meaning more than 3 times the eupnic tidal volume. So what was the receptor which was stimulated? It was our pulmonary stretch receptors, also known as slowly adapting receptors, which send the afferent impulse to the center through the vagus nerve. Our center is the integrating center is the medullary center, which is mainly the DRG. DRG which was firing is now inhibited. So once that is inhibited, the inhibitory signals are carried through efferent nerve. Efferent nerve is the spinal motor nerves. The phrenic nerve which supplies the diaphragm, and the intercostal nerves or the spinal nerve supplying the external intercostal muscles. So these are inhibited, meaning these muscles relax. So once they relax, the effect brought about is abrupt termination of inspiration, and there is prolongation of expiration. So what is the final outcome of this reflex? It prevents overinflation of the lungs. So we can see here, so the red line, whenever the lung volume is increasing, we can see activity in the vagal efferents, and the phrenic nerve activity is decreased, meaning the inspiratory signals are reduced. So we can see that compared to normal, the signals are reduced. So there is beginning of expiration, which is a passive process. So Herring-Brewer reflex we saw in normal physiological condition like exercise. So in pathological conditions like chronic obstructive pulmonary disease also, these stretch receptors play a role. How? So in obstructive disease, whenever the lung volume is increased, that stimulates these pulmonary stretch receptors. So we have the same reflex which is brought about through the vagal efferents, medullary center. We have efferent fibers through the spinal nerves, which acts on the muscles of inspiration. There is inhibition of inspiration or termination of inspiration, and increased duration of expiration. So there is a long, slow expiratory effort. So this is mostly important to minimize the dynamic activity and also to prevent the airway compression during expiration. So we always say that in COPD, it is very important to prevent this airway compression during expiration. So that is normally only maintained by our Herring-Brewer inflation reflex. So second type of Herring-Brewer reflex is Herring-Brewer deflation reflex, just the opposite of our inflation reflex. So whenever there's deflation of the lungs, there is fall in the tidal volume. So these receptors which were firing, reduce firing. So meaning there is decreased signal through the vagal efferents, integrated center. So whatever inhibition was there, there is decreased inhibition. Decreased inhibition or inhibition of inhibition is nothing but stimulation. So there is decreased inhibition of inspiratory signal, means the inspiratory signals, the inspiratory neurons are normally firing. DRG, which we discussed initially. So DRG continues to fire. There is an inspiratory ramp which causes muscles of inspiration to contract. So we have increase in the breathing. So whenever there's deflation of the lungs, there is increase in inspiration, or it brings about inspiration and also increases the rate of respiration. So if we have to trace this reflex arc, our stimulus for deflation reflex was marked deflation. Receptor stimulated is our pulmonary stretch receptors, and at times we have also the rapidly adapting receptors which are stimulated. Afferent carried to vagus, through vagus, relayed in the medullary center. Here now what happens is there is no inhibition. So the DRG continues to fire. We have the inspiratory switch is on. Impulses are carried through the spinal nerves. They act on the diaphragm. There's beginning of inspiration and inspiratory rate increases. So what is the final outcome? When the lungs are deflating, this reflex, it induces inspiration and prevents the collapse of the lungs. So physiologically, when does this Herring-Brewer reflex occurs? I'm sure like most of you are feeling this right now. Okay, that is during yawning. So whenever there is collapse of the smaller alveoli, there is a fall increase in the lung compliance. This causes the stimulation of our rapidly adapting receptors, which bring about increase beginning of inspiration and increases the depth of inspiration. So that is when we take a deep breath during yawning. So this reflexy open up all the collapsed alveoli. I'm sure everybody had one episode right now. So next, coming on to Herring-Brewer deflation reflex in a pathological condition like pneumothorax. So as you can see here, whenever there's collapse, it causes the distortion of the bronchial epithelium, which causes stimulation of our rapidly adapting receptors and stimulates inspiration. So whenever inspiration is stimulated, the depth also increases. This prevents the collapse of our alveoli. So what we have to observe here is, in inflation reflex, we had our slowly adapting receptors solely responsible, whereas in deflation reflex, we have slowly adapting as well as rapidly adapting receptors playing an important role. So to summarize the significance of the pulmonary stretch receptors, we have seen the structure, functions, properties. So their most important function is the Herring-Brewer reflex. We have already discussed enough about it. Apart from that, it also regulates expiratory muscle activation. It increases the parasympathetic tone, meaning causes bronchodilation and relaxation of the airway smooth muscle. It optimizes as a reciprocal relationship between dead space and our airway resistance, reduces the systemic vascular tone. And one important thing is it increases the heart rate by reflex cardio acceleration. So all these mechanisms, research is still going on. So we can just remember so many. But the Herring-Brewer reflex is being discussed in detail. So next is what the recent studies have been showing. So they have also known to control breathing pattern and also play a role in pathological conditions like asthma, emphysema, and pulmonary fibrosis. In asthma, there's increased sensitivity to normal stimuli. Emphysema, these pulmonary stretch receptors are responsible for altered breathing pattern. In pulmonary fibrosis, these receptors are responsible for altered lung compliance and rapid shallow breathing. They're also known to modulate air hunger and dyspnea. So these are the significance of our slowly adapting receptors. Next, in brief, I would like to tell about rapidly adapting receptors because they are sometimes known as rapidly adapting pulmonary stretch receptors. So they can be included under our topic of discussion for today. They were earlier called as irritant receptors. As I told you, they adapt quickly to stretch imposed on them, just like our touch receptors. So, this is an experimental evidence. As you can see, whenever there is sudden change in lung volume, the rapidly adapting receptors fire. After that, they get back to basal rate. Whereas our slowly adapting receptors, if the lungs are stretched, they fire, continue to fire. Whereas rapidly, only the rate of change of stretch, they respond. If there is sustained stretch, they do not respond. So location, I told you that this rapidly adapting receptors and C fibers are present in the airway mucosa, whereas our pulmonary stretch receptors or the slowly adapting receptors were present in the smooth muscle. This is present in the airway mucosa. So structure, they're again sensory nerve terminals of myelinated fibers. They are stimulated by sudden inflation, acute congestion, and inflammation. So they're known as irritant receptors, meaning they are mainly chemosensitive or respond to chemical stimuli like inflammatory mediators like bradykinin, histamine, prostaglandins, and also to irritation in the airways like smoke, dust, ammonia. So they mainly respond to rate of change, and whenever there is sustained volume change, the firing rapidly declines. So where are they significant? Mainly in pathological processes which involve irritation, allergy, congestion, inflammation, and they are proportional to lung compliance. They sense the compliance change. They are inactive during quiet breathing. Okay, so the stimulation of the central receptors produce excitatory response like coughing, gasping, increased mucus production, bronchospasm, whereas the deeply located receptors, they produce hyperapnea. Then we have the third type of receptor responsible for respiratory reflex, a C fiber ending. They are unmyelinated fibers present in the airway mucosa of alveoli and conducting airways. We have two types: pulmonary C fibers present adjacent to alveoli, they're known as J receptors or juxtacapillary receptors, and bronchial C fibers, which are present in the airway. J receptors play a role in J reflex. Okay, this was proposed by Pental. So whenever these J receptors are located between the capillary and the interstitial space between the capillary endothelium and the alveolar epithelium. So whenever there's fluid collection in this space, there is stimulation of this receptors. That is normally in physiological conditions like exercise at high altitude, pathological conditions like pulmonary congestion, pulmonary edema, or pneumonia. So once these receptors are stimulated, whenever there is stimulation of these J receptors, there is apnea followed by hyperventilation, bradycardia, and hypotension. So this is a triad: apnea followed by hyperventilation, bradycardia, and hypotension. So this basically tries to remove that whatever fluid or content was accumulated in the interstitial space. So whatever triggered this congestion, there's a reflex to relieve this congestion. So, okay, so we are almost coming to the end of it. Just to summarize, we have our involuntary or automatic breathing by the brainstem structures, that is the pons and the medulla, which is controlled by our cerebral cortex. When we want to voluntary control is when we are talking, when we are singing, when we are voluntarily hyperventilating, or during emotions. So there is cortical role in breathing. So we have the stimulus from respiratory receptors in the muscles, from the chest wall, chemoreceptors, mechanoreceptors, all these send impulses and regulate respiration. The key respiratory reflexes which play a role are the Herring-Brewer reflex, the deflation reflex, and the J receptors. To summarize, we have slowly adapting receptors and rapidly are myelinated receptors, whereas the C fibers are unmyelinated. The rapidly adapting receptors and the pulmonary C fibers are present in airway mucosa, whereas the slowly adapting is present in the muscles, smooth muscle. So slowly adapting play a role in Herring-Brewer reflex in they also increase heart rate by reflex cardio acceleration, so they cause tachycardia. Rapidly adapting receptors, it's mainly in the cough reflex, cause cough, bronchoconstriction. Unmyelinated C fibers or the J receptors, mainly they play a role whenever there is congestion or increase in the content in the interstitial fluid. They cause apnea followed by hyperventilation, hypotension, and bradycardia. So these are the various references for my study, predominantly books on medical physiology, few books on respiratory physiology, and a few journal articles. So I would like to thank our Dean and Medical Director of CDSIMER for all their support, and my senior colleagues, Dr. Prashant Kumar, Professor and Head of Department of Physiology, Dr. Veena, Associate Professor, who have given me a lot of ideas to bring out this presentation. I also thank my other colleagues, Dr. Shivmurthy, Dr. Kanchana, Dr. Chaitra, Dr. Ravi Kumar, and Rashmita for all the support. And special thanks to Dr. Puneet sir for giving this opportunity and always encouraging us. And thank you all viewers for making us a part of your day. So for all the pulmonologists who have been working in the front line all these days, our prayers for you. Stay safe, take care. Wish you all a very good luck. Thank you all.